Okay, Biology 12, it's time to finish off the last few notes on membranes. So let's get this done. If you go to the eCampus Live Biology 12 site, I'll just back up one frame here. The section that you would be at would be Topic 5, which is Chapter 4, Membrane Structure and Function. So we're going to finish this off. And the resources that you have here are the ones that'll show you about osmosis, diffusion, facilitated diffusion. This stuff down here in section 4 is really important. And in section 5, when we talk about using energy to pump things through the cell, largely we're looking more or less down here. Now, we'll get going with that. I want to get back to where we were in the notes. It would be at this point here. We were looking at passive transport, which was osmosis. And that's pretty important. Anytime you take a drink from a glass of water, for example, you're putting osmosis at play. And that is when you go down a concentration gradient. That's kind of natural, right? You drink the water and it flows into your cells. Your cells are more concentrated. So water, as I always said, goes where the action is. And this is pretty important. Your large intestine is responsible for a lot of your water absorption, and that's how your feces are formed. And your kidneys, if there's any excess water, will pick it up and use ultrafiltration to push it out into urine. Some of the excess water in your blood will come out as sweat, perspiration. Ladies, I guess we glow, don't we? All right. So you've got to watch out, because if you take on too much water, the amount of liquid, you could think of as the liquid substance of your blood, will increase, and that can increase your blood pressure, and that could be a real serious problem. That's the reason I put these pictures at the bottom, because it puts you at a greater risk for a heart attack or a stroke. Now, the stroke is a blood vessel that more or less pops in the brain, and the bleeding on the brain creates a lot of damage. It is reversible if you get to it right away. In some cases, I guess I should say. Paralysis of the left side of the body. My grandmother, for example, had a small stroke, and she was slurring her speech. You don't want to have too much pressure in your blood vessels. It can be a major problem. So we'll pop out of a little bit of talking about osmosis for a second, and what we should talk about are some active things. Now, your cell needs to bring in nutrition. Okay, so a Latin way of saying coming in is endo, and cyto means having to do with the cell. So the word at the top means bringing things into the cell. And the cell itself, I've stated on many occasions, is very concentrated. We're looking at a stockpiling of amino acids, of things like salt, a lot of sugars. Now, how do we get that all in? Because the way diffusion works is this should all spread out, but the cell becomes sort of a hoarder of things. Well, part of it is that the cell pumps it in. So we'll look at what we're talking about with endocytosis. Endocytosis would be, and I'll just pop out of here for a second, when a cell swallows something or brings something in. I've got a good animation for this. I'll just pop out for a second. Not those. Those are coming. There it is. And endocytosis is a very active process where the cell brings things in. And I think a really great example would be along the lines of white blood cells. In this form of active transport, the cell will literally bring things in. Cell eating, for example. So let's take a look at this. There's a couple of forms, 3 subforms. The cell can literally eat like a white blood cell would do. It can take in small amounts. So instead of phagocytosis, which is sort of large eating, it can do penocytosis. And sometimes it can do a form of bringing things in only once the correct lock has been activated on the cell. Let me show you. So phagocytosis is kind of interesting. If you think about an amoeba, for example, the cell membrane reaches up and drags in food. This is quite an exciting kind of active transport mechanism when you see it. So the outside of the cell is here and the inside is here. So the cell wants this, and it literally engulfs it. And if you remember pseudopods, they are defined as false feet. When you come through that membrane, what you see is that the membrane material became a bag for the food, and at that point it becomes a vacuole. Remember that vacuoles are just large containers of water or food that enter a cell. So a cell can actively pull something in. That's pretty interesting. When you look at something called penocytosis, known as cell drinking, this doesn't involve this sort of engulfing with pseudopods. Penocytosis is when small vesicles or I guess larger vacuoles, that's really the relationship. Vesicles are small and vacuoles are large. Pseudopods don't reach up and drag something in. So we'll take a peek at that. So if the cell wanted to bring in a particular fluid, it can bring it in and make a sort of an invagination or a pinch, and then the fluid just flows in. So you have a vesicle if it's small enough, or a vacuole. There's a third kind here where a receptor has to be sort of turned on before something comes in. And it's called receptor-mediated endocytosis. So there'll be these little, think of them as locks. And once they're activated, then all of a sudden there'll be endocytosis. This kind of reminds me of the little hairs, for example, on the Venus flytrap that you touch, and then the Venus flytrap closes over its prey. So these coated pits, think of them as little triggers. And we'll see here that specific binding brings something in. So you can think of it as a stimulus, if you will. Okay, so I'll let this play out. Give it a moment. So here's a substrate, and this little pit has these little receptors, and the receptor mediates the pulling in or the phagocytosis. That's an active process that took energy to do, but it was triggered, for example. And you know, when I think about this, growth hormone will act in this way, where cells will be told to take in all sorts of food, and the trigger is the growth hormone that's released by our bodies as we grow. And for example, a famous wrestler, Andre the Giant, produced way too much of it, and he just grew to be a tremendous size. That's a kind of endocytosis when the cell just keeps eating and eating and eating, and it grows really, really large. Okay, so I'm going to get rid of that tab. I've got all sorts of tabs for you here. We'll come back to some things as we go along. Just a moment. So endocytosis is a very active process. Okay. So when cells need to bring things in, they use energy. When cells need to export things, maybe they're hormones, maybe it's garbage, whatever it is, that can take energy too. And this is kind of an old picture, but what it's really designed to show is that cells can use exocytosis. So I'm sorry that this animation isn't working, but exocytosis, let's pop a little animation up for you. It's not really a hard thing to do. I'll just pop one up. Exocytosis is just the exact opposite of endocytosis. So let's see here what we've got. There's a couple here that I've looked at in the past, but I'm just going to mute that. So if the cell produced something, for example, protein in the endoplasmic reticulum, let's just say, maybe the cell produced a hormone, it needs to more or less send it out. So there is that protein going through the rough endoplasmic reticulum. I get it. And it goes to the Golgi complex for a little bit of packaging. This is a little bit of cell by. Once the Golgi has processed it, here it is traveling through, it forms this little vesicle. And when it leaves, it will head for the edge of the cell membrane. Let's see if I can just get that a little bit more, because it was, I think I skipped it, but here it is forming, and Let's see, we got a little bit more animation. Ah, there we go. And so it's received proper signals to leave the cell, and its membrane fuses and the contents go outside. So, for example, that could be something like insulin, which regulates your blood sugar. So, you could think of it as being released to the bloodstream, which would be exactly what insulin is for, right? That needs to be transported out throughout the body to tell our cells, hey, absorb that glucose and store it for a rainy day. So, exocytosis is just the opposite of endocytosis. Endo. And this is kind of neat. This says just click the image below. I have to update the slideshow. But what you see is the cell bringing in something to eat, to destroy, perhaps, if it's a white blood cell. So it's quite a neat little sort of process. A really interesting point, I'll go full screen here. So let me just hit play, is that membrane permeability. I told you that famous rule that like dissolves like. So you can use a little bit of white wine to help mop up a red wine stain. And some viruses have figured this out. That cover themselves in a little bit of the cell membrane. Lipids, okay, fat, oil, or wax, that's what the cell membrane is made of. So when a vacuole goes to leave, when it goes up to the edge of the membrane, it's made of a lipid outer layer, so it will merge with the cell membrane. So lipid materials can go through pretty quick, right? They just dock with the cell membrane, and the membrane gets really recycled a lot because things that come in become part of the membrane, and things that go out become part of the membrane. Eventually, you get the idea. Small molecules can go right through. Uncharged molecules and ones that aren't too really don't have a big charge can get through. When we look at I'll pop out for just a second. When we look at the proteins, this is a transmembrane protein. Some are channels that let things go all the way through. So I'll give you just a second because you're probably madly writing. But the membrane is supposed to be selectively permeable. So transport is important. Now, if you think, for example, this sort of brown protein that's going through the membrane, it's quite large. If, for example, water could go pretty well right through that. Really small ions could go right through, but large things could get stuck on the outside. For example, large carbohydrates, etc. Glucose is pretty big. It could get stuck on the outside. Maybe it needs some help to get through. So membrane permeability, well, if you wrap yourself in a lipid, you can get through the membrane faster. HIV is covered in lipids, and that's one way that it sort of integrates with the membrane of the T4 helper cell, which it goes after, and shuts down your immune system. But this is really sort of the four key things about the membrane that I wanted to tell you about. So I'm going to move ahead now. So I talked about these three forms of endocytosis, and they're really important. Phagocytosis, cells got to eat. Penocytosis, sometimes the cell has got to drink, bring in small amounts of liquids, etc. Phagocytosis, I think you should think of a white blood cell because when you watch it attack germs, it will literally reach out and grab them. And it uses, you remember those lysosomes? It will use those like weapons to literally blow holes in the cell wall of bacteria. It'll attack viruses in the same way. If you've ever seen the Magic School Bus when Ralphie gets infected, once the white blood cells know about the infection, they'll go right after it. And that receptor-mediated version of phagocytosis is really smart. Hormones tend to use that trick to trigger cells into doing what they need to do to differentiate. Girls are girls because of progesterone and estrogen. Boys are boys because their cells are taking up testosterone, right? You get the plan. Okay? So take a moment, scribble these down. This is what I would write on the top is endocytosis. I would just say active transport because it looks like it takes energy. It looks like a PE class with what those guys are doing. You know, it's right up there with push-ups. Okay. 3, 2, 1. I'm going to move forward. Okay. Make sure you get this down. So if we break it down to transport, I mean, I was talking about passive transport. We looked at diffusion in the classroom when I sprayed that perfume in the lab. We looked at when we did the dialysis tubing with the membrane. We saw that, you know, it was interesting that iodine got into the bag, didn't it? It stained the starch blue. That didn't take any energy, except for the energy of the particles just moving around. So we say that that's passive. So diffusion and osmosis are passive. You don't need any ATP. You don't need any energy. And facilitated transport, I'll just go up from 3 because we know about diffusion and osmosis. Facilitated transport is passive. So that's down a concentration gradient. I'm going to show you that one. Facilitated means somebody helps. Somebody helps the transport across the membrane without really requiring energy. That sounds like an interesting trick. Here's a hint. I'll show you the glucose transporter that does a great job on doing that. And then active transport. So at the top, you could write down endocytosis and phagocytosis and penocytosis, receptor-mediated endocytosis. They all require energy. You want to bring something across? That's fine. And it's usually either a protein, right, that's requires sort of a little bit of energy to pump something across the membrane. Got a little bit more to show you. Okay, so a little bit of yip-yapping, but I'll give you a chance. You've got your active transports, facilitated, and your diffusion osmoses. Okay? Okay, so facilitated diffusion and active transport. Kind of interesting when you look at these. Oops, went a little too fast. Let me bring those 3 points up. You need to move stuff across the membrane. So just pop out for 1 second here and zoom in. Now you need to get solutes or particly bits across the membrane. Here's some amino acids on the outside, and you've got to get them from, let's say, your digestive system, which has broken them down, into the inside of the cell. That's going to take a little bit of work. Sometimes there's little proteins in your membrane that will help that happen. Okay, whether or not it's active transport, this is like a little machine that uses energy to run and just pull things in. And sometimes there are little proteins that will just kind of flip when they fill up with things, and they'll just add things to the inside of the cell. Let's pop out for just a second. Okay, so I've got a few animations to show you here. Active transport. Let's see here. We've got osmosis, diffusion. Active transport, we already saw endocytosis, where that's where the cell literally just pulls something in, right? And there are a couple little forms there. If you look at an ion pump, sometimes the cell needs to pump ions in or out of the cell. And that's an active transport process. Now, it can get a little bit complicated here, but if you want to pump ions in, let's say that we have a little protein here, we can use a little bit of ATP, and we can pump hydrogen ions from a low to a high. Now, this isn't if you watch this, that's not diffusion. That's the exact opposite. That's stockpiling. So cells do have this little trick where they can use energy to stockpile ions. And this is, for example, was a hydrogen pump. And just move on to one more. Co-transport is kind of like getting into a nightclub with a friend. You're too young. You shouldn't be getting into nightclubs, but one day you will. You get in because of somebody else. So sucrose, which is a dimer, it can't really get into the cell. It needs, it's trying to sneak in. It can't get in. But due to the action of what's going on over here, right? There's a little hydrogen pump going on here. We just saw this a second ago. You see hydrogen sneak out there, or sneak through, and then sucrose went in. That was actually really subtle. Let's kind of watch that again. So sucrose is busy trying to get from the outside of the cell inside, so it's just as nice twice. It can't get through, except if it's got its buddy with it. So a little bit of hydrogen here got pumped out of the cell, and hydrogen sneaks through an open door, and oh look, sucrose goes in. That's co-transport. That's how sucrose actually gets in. It needs a little bit of hydrogen to help it sneak in. It took a little bit of energy to make that happen. So co-transport. Some molecules, to keep it simple, some molecules can't get into the cell without friends. Okay. I think the last one was endocytosis, which we already looked at. So kind of interesting. So three main sort of energetic ones. You can pump ions, you can sneak through the membrane with friends, or the cell can just eat or drink you. Ions, sneak in with friends, you're consumed by eating or drinking. Okay, so that takes energy to get you in. So now you know. There we go. I can close those now. So those are active transport mechanisms. There's one here called the sodium-potassium pump. Now, this comes up when we study nerves. I'm going to do you a very great favor. I've been meaning to do this for a little while. Sorry, folks. Slides 47 and 48. So this one here on the sodium-potassium pump, what I will tell you about it is that your nerve cells only fire when we pump the sodium and potassium levels, when we maintain them across the cell. And it says it creates a dipole inside the cell. Think of it as recharging a battery. Your nerve cells literally have electrical impulses because of the actions of like sodium and potassium. Now, normally we teach the sodium-potassium pump at this point in the course. I'm growing older and I'm growing wiser, and I'm just going to mention it right now. That's all I'm going to do. The sodium-potassium levels have to be actively transported and your nerves work. Okay, there's a neat little picture here about restoring those levels once a nerve impulse happens, but we're not doing it today. Because guess what? We're going to save it for the nervous system, where it belongs. So, I'm going to sneak past that for a second. Active transport, getting back to active transport, okay, it requires carrier proteins. And we said that the cell membrane is a phospholipid bilayer with some cholesterol for strength, and it's got these carrier proteins. And if you do active transport, you're going against the grain. You're going from low to high. That takes energy. You want to pump things? Hey, be my guest. It's going to take work. It's like pushing a car uphill. It's going to take some energy. But some things don't require energy. For example, let's pop out. We saw a couple of things. Basically, what we're talking about here is passive transport as a category. So when you look at things that don't require energy, right? When we look at osmosis, right, water flowing from a low to or from a high concentration to a low, down a gradient. Let that reload for just 1 second. Okay. Well, let's look at it this way. I'll get rid of that animation. It takes too long to load. But if you look at osmosis, water passing through a semi-permeable membrane, it tries to have about, and I'll just rewind that and run it again. You should have the same concentration of water on each side. These levels are about the same. It's at an equilibrium. Now, the only time it would not be at an equilibrium, folks, is if the water was going where the action is. Now watch this. I'm going to add salt into this side. Now, this side is salty. Well, these are some pretty attractive elements. And because they're charged, some water particles will try to stick to sodium, and some parts will try to stick to chlorine. Did you notice that when you add salt to one side, if there's a membrane here, the water particles can go back and forth. But when you add salt, more water particles actually came to this side. Well, that's because the water particles are attracted to these ions, and it will create a differential in height. And that didn't require energy. That's just pure attraction. That's passive transport. Okay? Osmosis is passive transport. Here's another one called facilitated diffusion. Now, you know when you go to the pool and they've got those water buckets, everybody likes to stand under the water buckets. And the water buckets fill up with water and then they face down, right? They turn upside down and you get soaked and the water hits you on the head. But those buckets only turn upside down when something fits into them, right? When they fill up with water and then they turn upside down. It doesn't really take any energy for the buckets themselves. The buckets just fill and they turn upside down. That's a lot like facilitated diffusion. Now, watch this. Okay, so let's go. I just have to sort of restart it, I guess. Let's see here. There we go. Okay, so let's say that this green ball is glucose. Okay, it enters the little protein. And do you see that? Now that was subtle. Because it fit in there, it changed the shape of the protein, and the protein, when it changed shape, just threw it in. It's called a conformational change. That's the big word for the water bucket filled up and turned inside out. Okay, it's just like the pool. Let's see if I can get it to run one more time. So this is how the glucose transporter works. Fills up with glucose. This channel, you just basically change the shape of the protein. Glucose goes in. Now, interestingly enough, this is just like osmosis or diffusion. It works down a concentration gradient. Nonetheless, it does help get glucose into the cell. And the last time I checked, cells run on glucose. Probably a good idea. So these little proteins just change shape when they touch glucose, and in goes glucose. Pretty cool. All right. I think I'll just close that little animation. We don't need it. So back to our notes. We'll just sort of polish this off. Facilitated diffusion, and I'm going to make a little change here, is from a low concentration to a high concentration. Okay? Glucose transporters. That makes it, it's a passive transport mechanism. So I had to change that, update that. That's okay. No problem for me. So your glucose transporter does this, and there's glucose sneaking in from the outside to the inside. Kind of looks fancy, but here's the molecule binding, and it just gets spat into the cell. Sounds to me like that's a good idea. When after this spits it in, kind of looks like a clothes peg, doesn't it? After it spits it in, it faces out like this. There you go. Glucose transporter, passive transport. Who would have thought it? Okay. It's not an ion channel. It doesn't take any ATP. It's really handy. Okay. These aren't just like pores in a membrane where things come through. And there's different ones, right? You've got glucose. A lot of these channels can be really specific to certain nutrients. So the cell has a lot of mechanisms to help it get a lot of things through. There's some, as soon as I mentioned this, I wanted to talk a little bit about these ion channels and the tetrodotoxin. If you look at a pufferfish, it produces this extremely powerful toxin. And what happens when you poison someone, like for example, cyanide basically poisons your mitochondria and they don't work. And if your mitochondria don't work, no energy, the cell doesn't work. Okay, so it's game over. When you look at a typical ion channel, for example, it would let things through. Okay? Tetrodotoxin, well, it plugs the ion channels. not a good thing. And things that would normally flow through the ion channels, right? Sodium, water, etc. Yeah, they don't get through. And that's it. Game over. Cell can't survive. So enough said. There was a lot in cell membranes for chapter 4. If you get a chance, by the way, sneak on over to cellsalive.com and check out some of these animations. Some really, really, really great stuff. So I'll kill that off there. No, I do not want to save that. So at this point, you say, okay, where am I at in the course? Where do I have to be? Well, let's go back one slide-a-rooney here, or one web page. So you've got your chapter 4 package. I gave it to everybody. Okay. And let's make it a little bit bigger here so we can all know it, love it, and live it. You want to get through working on this. It's going to ask you about active transport, passive transport. And this slide in particular, this section, please pay attention and ask yourself which side is hypertonic and which one is relatively hypotonic. What would the height of the water be? There's a little membrane here, and they're trying to ask you, hey, if you were the water, which way would you go? And which side would be higher than the other? Okay? Now, don't have some lame answer where you say, okay, 4 days later, it'd be like this. What we want to know is what would happen to the levels? Just period, without any sort of fanciness. This is a classic sort of provincial exam of old type question. So there's 4.3 asking you about osmosis and diffusion, right? Based on your lab. Some things that you have to draw in. Okay, fine, awesome, whatever. Nice little definitions, word search. So now that I've gone over this, hopefully I've given you enough to think about. And this brings us pretty well to the end of our chapter. So that means that we look like we have a test coming up. And I'm thinking probably Thursday of this week. You should have your stuff done. That leads us into chapter 6, enzymes, and we'll get going on the human body systems. And I think I'll mix up the order when I get down here. Take them in a slightly different order than previous years. Maybe we'll start with the reproductive system, for example. But that's it, ladies and gentlemen. And I hope you have a good day. It's Mr. B checking out. Now get to work. We'll see you soon.